So hi everyone, welcome to this week's edition of the ASAP webinar. My name's Anna, I'm from UNSW in Sydney, and it's my great pleasure to introduce this week's speaker. So before the introduction, I'd just like to add a disclaimer that we are recording today's talk. Just so that everyone knows, and it will be uploaded onto the ASAP YouTube channel in the coming days. For questions, if you could please put the questions into chat as usual, then we will help field the questions at the very end, and there'll be plenty of time for Q&A. And that chat box will not be uploaded onto YouTube. So, feel free to be candid, or don't be shy. Go ahead and ask those questions. It's my great pleasure to introduce Professor David Fernandez-Remolar today from the Macau University of Science and Technology. And his particular affiliation there is with the State Key Laboratory of Lunar and Planetary Sciences. So I know that's a mouthful. Sorry, I lost the window that I was doing the introduction from. So right now David is actually not in Macau. He is actually in France. So he's in sunny France. You can see the sunshine behind him. But when he's not in France, he's an associate professor at MUST SKL Planets. I don't know if that's what you call it for short. So David is a geobiologist who specializes in astrobiology, and he has 20 years of experience in searching for molecular and non-molecular biosignatures that are preserved in extreme environments considered as environmental analogues of Mars. So you can call him a Mars space explorer in a sense. So for these purposes, he's researched different analogues, including Atacama, Rio Tinto, New Zealand, Tunisia, and Alaska. So super exciting places on Earth that we all wish we could visit right now. And from visiting these places and from his analyses, he's developed and used very diverse sets of techniques involving mineral, geochemical, organic, and molecular analyses. So today he will be telling us about biosignature preservation in extreme terrestrial analogues of ancient Mars environments, which I'm sure also informs the search for life on Mars, of course. So without further ado, I give you David.
Okay, thank you very much for inviting me. It's a pleasure to be here. So first of all, I mean, as I haven't asked to describe my research, I can say you who I am. I mean, in regards to the research I do and what I do. So, well, I consider myself a geobiologist. My background is paleontology, sedimentary geology, but we have a professional experience on biosignatures in extreme environments. I am now mainly my current research is focused on long-term preservation of biomolecules in ancient salts that can be mineral analogues of different areas in Mars. I do some research on Mars geology, so I have a few papers on collaboration because I am not a Mars geologist, but providing my humble knowledge to that. So I try as much I can do is do some Mars geology research, because this is the data source for understanding which sort of extreme environments we need to research on Earth. I have a great interest on astrobiology of icy bodies as well. So indeed, some extreme environments of Earth can be used as terrestrial analogues of some icy body conditions. And also, in regard to that, I have a great interest in developing instrumentation to perform real-time environmental and biological activity monitoring. I mean, so to the end, this will be used for building maybe future tools for exploring Mars on other planetary bodies. Anyway, so first of all, a very short introduction that what are biosignatures, very informal. So what consider we biosignatures? Well, they are natural entities that are produced by biological activity, but they interplay with the biosphere and also with the hydrosphere. The important thing of the biosignature is that it encodes some biological information, if we know how to do that, obviously, and can potentially be transferred can be transferred in the geological record. We use, obviously, we study the information has been encoded in the geological record. So, and how is this information? Well, there are a huge list that I'm giving some examples. So this, as I am telling you, is very informal classification. For example, a pairs of minerals that which composition and structural relationship suggest biochemical cycles. Minerals formed under strong thermodynamical disequilibrium. Thermodynamically unstable compounds like gases, minerals, as I said before. Physical structures, I mean, spheroidal, ovoid-like, filamentous structures that can be supported by other geochemical or molecular evidences. This is very important. Set of organic series consisting with biochemical synthesis, large molecule compounds, etc. So the important thing is when you are studying a biosignature, you are not only looking at one structure, but the most important thing is you can combine different levels of information in regard to the composition, structure, etc. that provide enough information to have a robust biosignature that can be used, obviously, as a model for searching life on other planets. So this is an example. This is a toxin analysis. This is toxin analysis for that people doesn't know how it works. It's just an ion beam that is inside in the surface of the sample. And then what you can do is a map of the composition or molecular composition in the sample. So obviously, I will say that to study biosignature, in this case, this will be a very useful tool because it provides different levels of information. You can see, for example, here, a structure, there are some ovoids here that are associated to some arrays. And these structures are defined by the composition as well. You have, for example, here, you have, I don't know if it's seen, but a combination of different mass that are molecular masses, I mean, that are mapped in using this technique that correspond to the fragmentation of different phosphoglycerides, so or phospholipids. So this is, I mean, the way to study biosignatures, not only looking at one structure or one set of composition, but integrate different levels of information. Anyway, other important thing is, shall we use terrestrial biosignatures to look for life on other planets? That is a good question. So, what is the only information we have by now? We don't know by now how to define life. So the only thing we can go is to the information we are recovering in the extreme environment in regard to the whole, I mean, which are the main characteristics of life, and then to apply for planetary exploration. So this is obviously, we are assuming that life, at least in the solar system, is based in the same principle that Earth life. Obviously, Earth life, what does it need? Liquid water availability, molecules, and ions, energy support, and geochemical gradients. In this context, obviously, the terrestrial biosignatures should be the models that could be used to search for life in other planetary bodies. So this is what we have. We don't know for sure by now a good, or we don't have a good description or definition of life that could be used to explore any sort of life could be available or could be existing in the universe. So we have by now to use the terrestrial model. So in this regard, having this in mind, we use a concept that probably you will know very well, this potential habitability concept that help us to where to look. So, and this concept, what inherits is a potential habitability area is that area that has water, molecules and ions, energy support, etc., that support life. So having that in mind, another important thing, why an essential biosignatures for exploring other planetary bodies, and having in mind what I said you before about the terrestrial life can be used as a model for searching life in other planetary bodies, is all of this diagram. The important thing is that the potential habitat on Mars lets us know which terrestrial analogs we can use to look for the life traces of life, how they are preserved, how they are. And then the most important thing is go down to develop this information, help us to give enough information to create tools to search for this same of biosignatures in other planetary bodies like Mars. So this is more or less how operates the transfer of information to search for all life in other planetary bodies using the terrestrial life model in form of biosignatures. Anyway, so using all this information and looking at Mars, we have a very long list of areas that could be habitable on Mars. So I can give in a few examples, and then we will go to the terrestrial extreme environments that can provide the information of preservation of biosignatures that can be used going back to Mars for searching life on the planet. So, well, you have here several areas that are associated to extremely acidic or hyperacidic environments. Other areas are associated to geothermal activity, and some other areas like Terrasirenum, I will show you as well. They are associated to hyperarid environments, the producing of very specific salts. In this case, in the Citrus major case, this is an area that provides information about very deep regions of the Mars crust. I am not going to speak about that because it will take long in any case. So, in regard to hyperarid environments of on Mars, you can see that Osterloh in 28 in a science paper, this is 2010 JGR paper, plot out the different outcrops of chlorides, I mean the alloy sodium chloride. And you can see it's everywhere in the, nearly everywhere in the south highlands of Mars. So occurring at different altitude, elevations, and you can see the latitude is going from 0 to the south. So this is a Terrasirenum area. I'm sorry, I will show you where is that. You can see here this red square. This is an area I am currently studying now, okay? And you can see there are different areas with chlorides outcrops. So they are very abundant. They consist of about 5 to 20 meters thickness or thick deposits, and mostly they have this occurrence. I mean, they are associated to different streams that to the end, in ending places, you find the salty deposits. So it looks like we can see on Earth playa lake environments that are formed under parasaline on hyperarid conditions. So on the other set of environments is hyperacidic, as I told you before. The first evidence we had some evidence before, but before sending the Opportunity and Opportunity rover, this is the Barnes Cliff where they provide the definitive evidence for occurrence of jarosite and other ferric oxides that were formed very likely under hyperacidic environments. These hyperacidic environments not only occur in Meridiani, but also they are very, I mean, these salts are widely extended on Mars. So this is Aran Caus. Caus is a complex crater And you can see these air corridors. This has been formed by wind erosion. And we pay attention on this corridor. You can see a white band here. You can see here the scale is 10 kilometers. Thickness is about half kilometer, more or less. And again, we see that, well, the occurrence of ferric sulfates, and is not seen because the zoom application, but you can see that all this unit is made of nanophase or iron oxides, and you have here ferric sulfates that are evidence. They provide evidence that it was very likely formed under acidic conditions. So the acidic conditions were very extended in the planet. So probably if we have to look for life on Mars, we should focus also in para-acidic environments. And finally, other set of environments we can more or less understand they were working on operating on Mars is geothermal springs. So this is some evidence in Arabia Terra. In this case, you can see some very well cemented structures. There's a pedestal structure probably formed by the activity of geothermal spring enriched in silica. You can see some little tiny streams around that has been also preserved here. Anyway, so can we find these environments on Earth? And I can, obviously, you know that the answer is positive. Yes, we can. This is, I will show you some examples and how are the different biosignatures we can use for exploring Mars when they are used as models, biosignature models, I mean, for exploring this, the planet, for searching for life there. But before I am starting with the other, the different extreme environments having this listed that are found on Mars, obviously one environment that is now everywhere in this planet, they are cold environments because Mars is a very cold area, a very cold planet now. So for doing that, you have to go to very cold environments on Earth. In this case, one of them is the Alaskan permafrost. So this is analog of a Mars permafrost. So the point is here is what we can discuss about microbial preservation in permafrost. So, well, to reach these places, you have to use very unusual transport, so like hydroplane. So, and this also is reachable in the summer, mostly in summer. So you can see there still some snow there, but there is not. The lake is mostly frozen during the whole year. So what interesting thing here is when you are going an area, just you are not arrive and then collect the sample. You need to use several tools. I don't know if it's very clear. You can see here this is a drilling machine. So we that machine that allow us to reach the area where we could find the permafrost in this case. So I will show you this this tool in other areas as well. So anyway, so other tool we use is resistivity sounding. So this technique allows us to see how is the lithology in the subsurface until, let's say, 40 meters, 30, 40 meters. So knowing how is the lithological distribution in the subsurface or the physical distribution of materials in the subsurface, we can, operate in a more optimal way. So we can optimize the collection, sample collection. Anyway, when usually the permafrost in Alaska, in the North, in the area of North Alaska, is about 2.5 meters. So we run the resistivity sounding system. You can see that this is correspond this area corresponds to the very low or medium low medium high resistivity or medium low conductivity. That's in the inverse of conductivity. That will correspond to the frozen place. This dotted area is it looks probably correspond to a rocky material. Anyway, so you can see that it's true that the permafrost more or less appear below 2.5 meters. Interestingly, well, this is the the aspect of the samples we collect. It's pure ice that is preserving a lot of organic compounds. I don't expect to find this on Mars, but what probably we will find on Mars, it would be a dirty ice, trapping different sort of materials. Anyway, the interesting thing is that you can see the abundance of cells, per grain, going from less than 1 meter to below to 3.6 meters, and doing the growth media. Growing different samples using different media. So we see that at even 3.6 meters, there are positive results growing in microorganisms and very different microorganisms. So what it says that the preservation in the ice is very good, even at the level of microbes. I don't know, obviously, the exact age of this permafrost. Obviously, it's not very recent, but if we go out to Mars, the age of the permafrost is much older. So I don't know at this point if we will be able to preserve living cells. Or cells that could be, are in standby and could be living after applying different growth media. But for sure, we will find different sort of long biomolecules that could be detected using other techniques. Anyway, so other set of environments I was listing on Mars is geothermal systems. This is, in this case, I can show you geothermal systems that feed surface water bodies in New Zealand, and when I was visiting the area. So the most characteristic of these springs are strongly acidic. So you can see the pH is below 2.5 and the temperature is very high. So even in these conditions, if you analyze the samples that were taken from this area, from this sampling points, that you can see that the pH is very low and temperature is high, we have a high diversity of molecules. We didn't grow anything here because we weren't able. We didn't have the techniques to be done in situ, but we collect samples, water samples, and we send to a high resolution mass spectrometer that can provide information about the sort of different biomolecules you can find. And you can see it's highly diverse, even in this sample point that has a 1.5 pH and the temperature was very high. There are a very diverse of biomolecules that saying that probably they are microbes, they are living, and this is in relation to a high diversity of a biomolecular record. So other sort of environments we are discussing is pyrite, and remember that one of them was the showing on Mars is those that produce halide, sodium chloride. So this is short algerite. There are different pyrite environments on Earth. As you can see, this picture is just a basin, saline basin that is seasonally filled by water that obviously will represent a very extreme conditions as can be found on Mars. These areas can be used, for example, for testing some instruments and collecting some samples, for understanding who is living there. But if I show you another picture, in this case the Atacama Desert, you can see that if I say this is coming from Mars, you will say, yes, this is a picture has been taken by curiosity or opportunity, but this is Atacama Desert. This is the Salar Grande area. Okay, what is very interesting Atacama Desert? Well, the most important thing is we can describe Mars as a volcanic planet that is associated with a lot of salts. So, as you can see in Atacama, you can see how these salty bodies are formed in the lacustrine, obviously lacustrine areas, but are fed by the hydrothermal activity that is promoted by the volcanic activity. You have the geothermal activity and then the salts produced by this activity. And you have modern systems, but in the case I am interested, you have fossil systems. In this case, is the Salar Grande area, Salar Grande fossil lake. So we can see how is preserved the biological activity in the last 100 to 200,000 years. So this is some pictures. You can see how is some Google Earth, and the other one is a picture was taken by myself. Some drilling places we will where we were collecting samples. You can hear this is the central basin. You have here a fault that is a living fault. So you have a lot of seismometers here because there are a lot of earthquakes in this region. And this fault has, split up the basin to produce a minor basin that also salts were produced. Interestingly, when we went to the central basin, this is more obvious area to operate, it was complicated to drill it because we were not using drilling fluid. The drilling fluid has a problem that can contaminate the samples. So we decided not to use drilling fluid. But going to another place, in this case, there were a combination of allied with some detrital that provide a better area for penetrating the subsurface and collecting samples. So it's the reason we decide to work in this place and then to try to collect as much samples as we could. So this is the central basin in the Salar Grande. You can see its scarp has about 5 meters. So you have at least 3 scarps here, so about 15 meters. So it's very interesting because fits well more or less to the chloride deposit we find in Terra Sirenum, as I have told you before. So as you can see, this picture is a pure alite. So has very small amounts of sulfates, but 90 to 99% of the composition is chloride. Okay, other source of samples were provided by drilling material that were obtained by the different mining companies there. So the interesting point here is you can see different drilling places, different sedimentary successions. You can see that the lake is filled by 2 different main sedimentary bodies, and the lower one is detrital cemented with sulfates, and the other one is pure alite. So they are 2 different. This is important so you can understand better. We are discussing later. So when you are looking at the different sort of minerals we found, one was oxalate, and weatherite is an oxalate here. You can see here oxalate here as weatherite. We found oxalates here as an ion compound. I mean ionic compound. This was determined using ion chromatograph. Ion chromatography. I have more oxalates here. So we have this hexagon. So where you can you find no, sorry, this is not one. Is the sorry, the triangle. You can see where the different oxalate appear. So interestingly, a few years ago, oxalate was found on Mars. So from this finding, you have the question: this oxalate is biological or is just produced by some chemical processes. So we try to solve this problem using Atacama Desert. This picture is to show you one important aspect of the Atacama Desert. It's a very dry environment, but you have a source of water that is this mist. This is Camanchaca. Camanchaca is a mist coming from the Pacific Ocean and entered by noon in the afternoon into the desert. That means that there are some sort of water that can be used by biological activity. Okay, so if we look at some stones, we realize there are some lichens. There are lichens everywhere in Atacama Desert. So when you are listening that Atacama Desert is the driest place where no life can emerge, that is not true, because you can find this sort of living forms that they are operating there, and they produce obviously different compounds that can be detected using different techniques. Okay, this is one of the sections I showed you before. I was confused before about triangles and triangles and hexagons. In this case, now oxalates are marked by hexagons. You can see here that some oxalates will occur in the detrital place, and other oxalates occur not in this case in the pure alite material. So plotting this this occurrence in a diagram against sulfate and chloride, we saw that there are two different sources of oxalates, looks like. So we can say that there are some detrital oxalates that, that are mainly characterized by using XRD, and other oxalates that only characterize using ion chromatography. So in this, this fits well, but the occurrence of two different pathways for forming oxalates. That is, one is produced by the activity of living forms like lichens, and bacteria obviously can be produced. And then they are produced as biominerals that are are not soluble in the when they are exposed to some water activity, and they occur as detrital minerals. On the other hand, we have another source of oxalates that are found in the pure alite member. Let's say that this upper unit that are only formed by sodium chloride, that are only detected by ion chromatograph because they are very small amounts and are produced by extreme oxidation of any biological product. So probably the Mars oxalate comes from the degradation of different organic compounds that are exposed to high solar radiation and UV radiation. So very likely the oxalates we find on Mars is produced by no biological activity. So they are abiotic process that produce these oxalates. So this important thing that biosignatures on Earth can also use to say whether or not we can find one potential biosignature on Mars is truly a biosignature or is not. Anyway, so in this area also we didn't go for oxalates. We use the oxalates we found using different techniques just because we saw an opportunity to test the sources for oxalates on Mars. But we went for looking for microbial activity, or let's say traces of microbial activity in the ancient salts in Atacama. So doing that is the same thing. We first of all use different techniques for sounding the subsurface. This case was the same thing, resistivity sounding. You can see we found different lithological units, and we went for the ring one because it was a salty deposit, but the lower one is a blue one. This is also a salty deposit, but probably was wet salty deposit. That means that probably is a deposit has been contaminated but recent water activity. So this is coming from the subsurface. So we decide to go to the green one. The upper one is it has a high resistivity. Oops, sorry. And we discarded about sampling and also analyzing as we did with the green member. Anyway, well, obviously when you do a drill, you obtain cores, what you do is a stratigraphical section to see how different parameters you are you're taking from analysis. These parameters obviously it can be microbial or molecular traces are distributed in the around along the depth. So anyway, so the other technique for collecting samples you can see is a drilling machine. As you can see, we didn't use drilling water so to prevent contamination, and this was very important because you don't use the vacuum. You couldn't move downwards because the drilling system stuck. You couldn't drill. So using both techniques, you can see we obtained a very nice core, a non-contaminated core that was sent to be analyzed by using this instrument. It was indeed an instrument developed by Center of Astrobiology in Madrid. That is, as the name is solid, sign of light detector is based on the association between antibody and antigen-antibody, close association. So obviously using different antibodies, you could detect the presence of different compounds. I mean, when I mean compounds is biological compounds that could be present in the subsurface. And this is the result you can obtain using all these techniques. You have depth and also intensity, occurrence of ausence of different compounds that are associated to a specific or general groups of microorganisms. And you can see also the intensity of, the intensity can give you more idea of the abundance of these different, let's say, biosignatures. Okay, using different, very simple mathematical or techniques, you can understand how the preservation process can occur in these salts. Going from the using the total relative fluorescence against the Simpson index, that is an ecological, an index used for understand the ecological diversity of a given area, that is very useful for this sort of analysis. You can distinguish different sort of materials based on the composition of different antigens. So you can go for allied reads. Whoops, I cannot move, sorry, I cannot move sometimes the arrow without moving to the next slide. Well, you can see that the highest diversity with the highest fluorescence occurs in the allied deposits. That means that allied is a very good material to preserve or biological compounds, but also at more or less the same level, again, you see that an anetrate and perchlorate member, they were, in deeper areas that are below the allied deposits. You can see that the preservation is, you have a very high diversity that also is associated to the high preservation. Interestingly, the lowest diversity and lowest fluorescence was given mainly by the upper deposits that correspond to some detrital and saline that are, exposed to the surface. So you can see more, you can have a sense how the preservation operates in these materials. So this, you can use this information for looking for traces of life on Mars, on other sort of different environmental also parameters. Is I will say that allied and also salty materials like perchlorates and nitrates are very good for preserving this sort of record. And if we run the analysis in the other side, I mean, using the different antigens, you see a very interesting thing here, that antigens that provide information about the occurrence of lipids is very close to those antigens that are associated to nucleic acids. That means that the preservation degree in salts is very good. So in addition to find different, let's say, biosignatures, we also have a sense of the preservation potential of different materials in this area, and which can be applied for, searching for life on Mars. Anyway, so let's go to another environment I have listed on Mars, and this is acidic environments. So most of my experience comes from this system. So what is interesting Rio Tinto? Well, it has, as I told you, is an hyperacidic environment. The pH is between 1 to 3, but also because it has a very high concentration on iron, as we can see in Arancaus or in Meridiani. So also another important thing is a highly oxidizing environment. So potentially, I will say that you cannot find biomolecules or organic compounds in the geological record of this environment. But this is something I am saying now. So let's say it is true, because most of the planetary researchers, they consider acidic environments as not good for preserving biosignatures in form of biomolecules, for example. Well, anyway, let's go. Oh, sorry. Okay, well, this is a picture of one area, sampling area in Rio Tinto. This is Berrocal. One interesting thing here is that at the first side, we can see some corrugated structures that correspond indeed, so probably to mineralization of different streamers. So, but not only in the modern environment, but also in the ancient environment, in the fossil record, we can see in the geological record of this environment, you can see also different evidences showing that the preservation is good, but not only for biological structures, but also for biomolecules, as I will show you later. Anyway, how Rio Tinto works is the acidity comes from meteoric water percolating an area that is enriched in iron sulfides. There are microbes that use this mineral as an energy, and then the byproduct is a solution enriched in sulfate, ferric ion, and also a high concentration in protons. You have the full reaction here. The interesting thing in Rio Tinto is not only that obviously the environment is acidic, but the environment is very stable because it's controlled by a ferric buffer. So even in very wet conditions, on very dry conditions, the pH is stable. And no matters if it's extremely acidic or extremely alkaline. If you have an environment that is highly stable, you have a high diversity in microbial life. And this has happened in Rio Tinto. Anyway, so other interesting thing of Rio Tinto, I mean, this picture is not very good, this slide is not very good, is the occurrence of different iron oxide deposits that show you that Rio Tinto has been operating about, at least up to 30 million of years ago. So you have different time slices where you can collect them, to explore them, and collect samples from these tiny slices that has been formed in form of different terraces, and then to see how the different biosignatures are preserved. So you can see how preservation goes in a time arrow that goes up to about 30 million of years. So in this, you can see here, this is the oldest terrace in Rio Tinto, has between 6 to 2.2 million of years. That, well, you can find this sort of structures that indeed show you that there are microbial activity operating over operating there about this time. Anyway, okay, the first thing we can find in terms of mineral biosignatures is some carbonates we found there. This is a SEM picture. This is an SEM picture of a modern sediment. So when we were looking at the SEM images, we realized that we found some minor structures or minor laminas that were enriched in carbon and ferric iron with oxygen. So that could be associated to the formation of ferrous carbonates, as this is using the SEM of some evidence of the occurrence of these minerals. But it's interesting because can you expect to find carbonates in a pH or at a pH of 2 or 1.5? So using different techniques, not only the SEM, including XRD, X-rays for mineral determination, we found also ankerite. And analyzing these sediments using this technique and going to ancient deposits, that we also found these sort of materials. I mean, you can have carbon, oxygen, and iron. Probably this in this case won't be ankerite. Ankerite is a ferrous carbonate with calcium and other cations. In this case, this will be other carbonate that's made of only by iron and iron. I mean, the only ion is iron made of. So we saw that the preservation on the time is up to, because this sample comes from 20 to 30 million of years, is very high. So, but we are sure that they were produced by microbes, because obviously they could be produced by obviously abiotic processes. It's not very frequent on Earth, that sort of these materials are produced by abiotic process, because there are a lot of living forms. But obviously, to know if it's a good biosignature, you have to test that using another technique that is isotopic analysis. We found that this carbonate, not the whole sample, but exactly the carbonate, has the fractionation that fits very well by microbial activity. So carbonates are formed in acidic condition by microbial activity, and this is very important. So this is a clear example of one metastable mineral that is formed in acidic in a condition that you don't expect to find it. But as there are microbial activity, they are produced, and they can be used as a potential good biosignature. Anyway, so not only minerals, but also some biological structures are very well preserved in the ancient deposits of Rio Tinto. Well, sorry. These are filaments, microbial filaments. You can see their branches here that are embedded in an iron oxide deposit that has about 25, 23 million years. This is the upper part of the geological record in Rio Tinto. But also you have a very well preserved, a very high detail deposits of microorganisms. You have plants, you have insects. Here are insects. That is not very clear, probably are plants. Using the SEM, you can see that the quality of preservation is very high. That is, this sample comes, and the sample I showed you before also comes from the 2.2 million of year terrace. The quality of preservation is very high. Even you can see probably a structure correspond to bacteria. But as I told you before, the geological, the planetary geologists and other geobiologists consider that oxidizing conditions are not good for preserving biomolecules or organic compounds. You can see the organics are very difficult to find, not found. Their sense can be attributed to the low preservation potential of the compound. This was in 2004. And then they say that better targets for the in situ search for ancient organic compounds on Mars include clay-rich deposits due to the ability of many clay minerals of Western organism inhibition decay. So the exploration of Mars is focusing now in fine-grained deposits. That is very good. They trap organics very well and they can preserve them. But it's true then that the organic compost doesn't preserve in acidic conditions. Well, doing the same sort of analysis in the, for example, this is the old terrace between 6 to 0.1 million of years. You can see that the results are very, conclusive. So you have a lot of compounds that are preserved in these rows. Indeed, you can see how these molecules are mapped in form of, in some cases, of filaments. There could be here a boundary between two different set of filaments. Anyway, so by now you can see that the preservation under in acidic and highly oxidizing condition is very good. So if we are going to the upper part of the, or the, let's say, lower part in time of the geological record in Rio Tinto, the geological record of this acidic environment, this is the upper Gaussian unit, about 23. People say, other people say it's 30, 29. I am going always to the youngest, estimation of the age of the units in any case. So, well, you can see how the rocks are massive iron oxides, as you know. So you look in the using a thin section in the microscope, and that's full of obviously biological structures, as we can see in the youngest terra, the oldest terras that have about 6 to 2.2 million of years. You have the same thing, very well preserved structures. But using the spectral analysis, in this case TOF seams, you can see that they are preserved, they are associated to the preservation of large molecules. In this case, they are phospholipids. Indeed, looking at other areas of this sample, you can move like a microscope using this technique, the TOF seams. Well, you see there are a high diversity in biological structures, but also that are associated to large molecules. In this case, this structure looks like they are associated mainly to ceramides. They are nitrogen-bearing lipids, and also other ones, ceramides, sorry, and DIG. They are glycerides, diacyl glycerides. So if you combine the images, you can see they are associated, they are probably different, the preservation of different sort of biofilms. One enriched in phospholipids, another one enriches in ceramides, in this case, sphingolipids, sorry. So some of them could be even glucose ceramides. So, but even if we use more resolutive analysis to search for compounds, biological compounds in these rocks, looking at the oldest terrace, remember that Gaussian is not a terrace, but it's the oldest record. Gaussian has 25 million of years. The oldest terrace in Rio Tinto has about 6 to 2.2 million of years. We were able to find peptide chains. So, and you can see, well, you can think maybe it's contamination, but when you run the analysis using youngest rocks and going to the oldest terrace deposits, in Rio Tinto, you have three levels of terraces. You have a young terrace about 4,000 years. You have an intermediate terrace about 40,000 years, and all the old terrace, as told you before, between 2.2 to 6 million of years. The contamination couldn't produce this because, the intensity decreased with age. And indeed, the sequences, some of them fit well the microbes you can find there, like Salmonella. You find obviously Homo sapiens, but Homo sapiens, this sequence is very general, as you can see here, because Homo sapiens can also, this sequence, protein sequence can be associated, occur also in different plant species, like the sugar. Sorry, I didn't explain very well this technique, but that is, high resolution mass spectrometry that you use to sequence peptide chains. I mean, so the fractionation pattern gives you the sequence, the peptide sequence, or the sequence in the peptidic chain, sorry, sequence of amino acids in the peptide chain. So, and finally, not only in the surface, but also in the subsurface, Rio Tinto, you find a lot of biosignatures as well. So, well, the same thing, but at a bigger scale, for looking at an area where you want to collect good samples, you have to do sounding. This is also, resistivity sounding, but a higher depth. So using this technique, well, you can run different, you can find different targets for drilling, and then you start your drilling. And then you, what you are looking for using this technique is two main parameters for looking for life in any place: is water and energy sources. In this case, it will be mineral sources. In this, you find this in areas with lower resistivity. Water, using these two parameters and using the resistivity technique, we find, as I told you, a couple of drilling areas where water, as you can see, water is found not, the water itself, but you can see how altered is the rock. That means that it's in permanent contact with water, but also we found in the same drilling site, in this case, it's BH10, mineral source for energy, as pyrite. Running microbial analysis, and this is using different stain techniques. So with some different microbiologists found even not only isolated microbes, but also biofilms. So, biofilm is a serious thing about talking about or speaking about biomass. So this microbial activity surely has to give some imprint in the geological record, and it does. Well, these are the different drilling cores we obtain. I mean, represented in form of stratigraphic successions, so stratigraphic columns. So the BH10 and BH11 was performed by the SON ERC proposal, and BH4 and BH8 was performed doing in a NASA proposal. So we have different sources of information, to study the subsurface in this area. Anyway, so obviously, this microbial activity, as I show you in form of biofilm in this case, obviously provides an imprint in the geological record. That's, you can see in this case is form of minerals. You have barite. Barite is an hydrothermal mineral. Let you know that obviously the Rio Tinto basement contains rocks formed under hydrothermal conditions. Barite, about 200 million of years ago, 200 something million years ago in Carboniferous, and barite is a mineral formed by hydrothermal activity that is obviously abiotically produced. But you can see these structures that are formed by, as I showed you before, carbonates. So there are some ways that microbes, reduce sulfate through, oxidizing organic matter that producing then carbonate. So in this case, this will be another way to say that this is a good, biosignature, mineral biosignature to apply in the subsurface and other region, including maybe Mars. Obviously, what I will do in addition is to search for the isotopic composition of these carbonates, but obviously we don't have the time to do it. In any case, as we have the model has been, found before in the modern sediments and in the old sediments in the Gaussian, as I showed you before, I mean, the formation of iron carbonates, and showing that they have also isotopic fractionation of carbon that agrees with the microbial activity. I have no doubts that they were formed by microbial activity as well. And we have microbes that we know in the subsurface that can run this sort of metabolic pathways. Anyway, other interesting thing is not only we found mineral structures, but also structures that they are consistent with microbial activity as filaments. These filaments, you can see, are enriched in nitrogen. So this sort of microbial remains could be associated to the nitrogen cycle. Using the topsin, in this case, we also saw that in this sample, this is the same sample that we see here. Here. This is the same sample. This we see different species of nitrogen, inorganic nitrogen, going from ammonia to nitrate. So you can see that probably this is consistent with some nitrogen bacteria that has been fossilized in the subsurface. Anyway, not only provides information about the metabolic pathways, but also who is there. And who is being preserved there. Sorry. So in short, I am saying show you this mapping. You can see just a dot there, that is a bright dot, that means an intensity in this sort of ions that correspond to phosphoglycerides, or phospholipids, sorry. In this, these phospholipids are associated also, you can see here in the negative ion mode, you can see that have the same structure, so a high intensity in fatty acids. So this is probably palmitic and other fatty acid. So fractionation of in mass spectrometer of the fatty acids we know comes from these phospholipids, but not from the ceramides. So we can see the other group of lipids here that has been preserved in the subsurface. This is this sample was collected about 40 meters deep. So anyway, we can start using also distinguish different sort of microbial forms in the subsurface using spectral techniques to determine this sort of biosignatures. And well, this is my last slides. Sorry for being very messy, but I have also some problems with the computer to prepare this talk. We are all the environments I have discussing are not indeed all. So they are about Miocene, Oligocene, up to 30 million of years. I am now focusing on studying salts that are, much more older, but this is not the case to discuss here. So, but we should look at long-term preservation traces, of traces of life, or microbial activity, because obviously Mars is a very old planet, like the same, or more or less the same age as Earth. But I mean, when I say this old planet, is the geological record correspond to very old deposits. I mean, much older than 3.2 billion of years. So one thing we should also do is look at biosignatures in very ancient materials on Earth. This is the Komati River. If there is some geologists there, so probably will know this name will won't be uncommon. Anyway, so this is 3.3 billion of years and shared. And there also you find traces of life. You can study using this sort of techniques, and it is Kronberg formation. This is the thin section I showed you before. And you see that there are traces of organic compounds that can provide information about long-term preservation in a very active condition. So probably the preservation degree on Mars will be higher if we go to the rocks to collect rocks of the same age. Well, this is all I wanted to talk. I am not discussing white salts. Probably from the discussion in this in my talk is they are very preserving mineral matrix for traces of life. Anyway, I was told to show this. This is about some sort of course about of astrobiology, and well, I suppose you are very welcome to join that. Anyway, that was my talk.
Okay. Great, thank you so much, David. Yeah, so the last slide he presented is just a plug for next week's ASAP talk, so I'll send an email out about that shortly. So we have some questions. I'll field those questions, and I must say, for those who need to go, feel free to go, but those who want to stay and chat, we can stay here and chat. So yeah, the first question is from Andre. Do you want to just unmute yourself and ask?
Sure.
First two questions.
Yeah. Two things that I wanted to ask. Well, first of all, thanks for the nice overview and the nice webinar. I thought it was really interesting that you mentioned the halite deposits, the halite outcrops in Mars. It seemed like they were restricted to a specific latitude range. Why is that? I was curious.
Yeah, it's a good question. Well, I mean, the north regions of Mars. Well, I am not a Mars geologist, but I think these are the answers I can give you. Is much modern, much more recent. So probably if they are salt deposits, they are covered by more recent deposits. That is the one thing answer. The other thing, the other answer could be that most of these areas, I mean the south areas of Mars, are a lot of different kind of volcanoes buildings. So maybe it could be also associated to the volcanic activity. This is because Mars is high, or the subsurface Mars, or the crust and Mars mantle is enriched in chlorides. So probably the volcanic activity, favor the release of chlorides by volcano, obviously, to the atmosphere, and this and also to the hydrosphere. So probably, this favor the formation of water bodies that were enriched as well in chlorides.
Okay, thank you. The second question that I had is, you mentioned the need and the way that we currently center our analysis is always, of course, based on what we see on our planet, and it's a little bit centered on the examples that we have. And so I understand that big focus is centered on terrestrial-like biosignatures. I was wondering if researchers are also looking into any potential non-terrestrial ones, as in like trying to sort of more speculative of alternative biosignatures that aren't necessarily Earth-centered or Earth-based.
Well, I cannot give you an answer to that. So as I have been working all my life with terrestrial environments, I have been focused on that. I know that there are people working on theoretical biology that trying to answer the question, what is life? And then from this perspective to go down to obtain more practical, results, and to see whether we can form different traces of life based on other, let's say sort of life, no, or kind of life. So I cannot answer that question really.
Okay, thank you.
No, you are very welcome.
Thank you very much. So the next question we have is from Yan Yuan He. I don't know Yes. Would you like to ask the question directly? Yeah, or okay. I will just ask it. What methods could we use to understand how long organic compounds have been preserved in the Rio Tinto samples, or potentially could be preserved in the Rio Tinto samples?
Which sort of techniques?
Yeah, like how do you
Okay.
figure out how old things are or how long things have been preserved for?
You mean, I am confused about the question. Probably he is referring to age, or I mean, how to determine the age, or how which instrumentation is used to find biosignatures in the oldest deposits of Rio Tinto. But I can answer both questions in any case. We use, it's not easy to obtain the age of the ancient deposits of Rio Tinto, because the most accurate, techniques is radioactive, radiogenic isotopes. The oldest deposits are lacking of that. So, up to 6 million of years, you have some isotopes that can be used for aging the rocks. So, so we have to think about how to do that in the future. That's a good question. So, assuming that we have the techniques for aging rocks, the techniques we are using for obtaining, biosignatures are quite different. You are going from different mineral techniques, inorganic, chemical inorganic techniques, ion chromatography, to spectral techniques, mass spectrometry. So this wide is highly diverse. So it's so big, suite of different instruments that, I mean, I could not list of all of them. Any instrument can be useful for, studying rocks in Rio Tinto. I mean, any to obtain, compounds and also minerals, structures, etc., for studying Rio Tinto.
Okay, so got a note from Miriam that apologies for not having a microphone, but yeah, thank you for that comprehensive response. If you want to ask any follow-up, yep, they say thank you.
It's okay.
Cool. So next question is a quick one just from myself. So you showed a lot of mass spec results for palmitic acid or palmitate. And I'm assuming oleic acid, which should be quite abundant, isn't as visible because it gets oxidized over time. So do you see those oxidation fragments for oleic acid, or is it just like totally gone?
Well, I haven't seen any of them, so
yeah.
I don't know. Probably because we don't have enough signal of these sort of acids,
Yeah.
So what I can say you, the preservation in Rio Tinto is very good, and it's because the mineralization is very fast. Mineralization inhibits microbial activity. That is the main factor to degrade organics.
Yeah.
So I mean, if you are using the right technique, you will find nearly everything. Obviously, except in DNA. I won't say that DNA is something you can find, or who knows. But anyway.
Okay, fantastic. Thank you. No, it's really impressive, actually. Next question is from Maggie. So this is a general question, and Maggie, you can just unmute yourself and ask if you want. Okay, I will read it out. She's in a noisy environment. So the preserved organics have likely undergone diagenesis or have been subjected to microbial degradation. So for organics to be preserved in the rocks and dated by lithology, how can you tell when the organics are preserved? And what is the mechanism for the signature functional groups or structures to be preserved for so long?
Well, I think I discussed before that. So for sure, the organics and biomolecules in the fragments of biomolecules are preserved. I don't know if he or she is referring to the Rio Tinto. In salts, it's quite evident. You have in chlorides, for example, you have rocks that are preserving DNA in an age up to 4,000 to 400 to 500 million of years. So there are people that are extracting DNA from these rocks. And in Rio Tinto, you don't have DNA for sure, but you have other sort of organics that were very well preserved, and is more or less the mechanism is the same thing. High mineralization, very fast, preventing microbial degradation of microbial degradation by microbial, or degradation of biomolecules by microbial activity, because when you have a solution as highly mineralized or it's very fast mineralized, you decrease very much the water activity. Water activity is a critical factor for microbial activity. So this is very important for Mars, because Mars has been, estimated that for the same solutions, the water activity will be even lower. So, I mean, lower than, around 0.5. For people that are studying water activity and microbial activity, can understand it's very low. So I think this is one essential factor. I mean, the water availability that is restricted at very high speed, because the mineralization is very high.
Okay. Yeah, I think that is a pretty comprehensive answer, and it gives a really clear idea. And Maggie adds that, thank you. And that also means that water activity has to remain low for millions of years for that preservation.
Obviously, in a mineral matrix, I cannot tell you for a mineral matrix, because I haven't seen any paper about that. But for example, for this ancient resin, amber. For
Okay.
amber, the amber has been extracted bacterial spores. I mean, and the water activity, so that means that preservation is very good,
Yeah.
Then the water activity was lower than 0.6, 0.5. So that means that the water activity is very low. I assume that for allied, for example, not fluid inclusion. Fluid inclusion means water, but for the mineral matrix in allied, should have the same water activity, more or less, the same more or less, same degree.
Yeah, it's pretty mind-blowing. Yeah, very, very impressive results. So I think that's last call for questions for everyone. If not, thank you so much, David. It's wonderful results and amazing pictures.
Okay.
But yeah, thank you so much for taking the time to speak to us from halfway across the world from Asia. We really appreciate it. And yeah, all the best.
Okay, thank you very much. Well, hopefully see you all soon.
Great, thank you.
All of you.
Thanks.
Okay, bye-bye.
Bye-bye.
Bye.
Thank you, bye-bye.
And thanks, Hannah, for hosting.
My pleasure, thank you.